Okay, so Stephen and Susanna contacted me about, right after the deadline and said, we got a submission for the PGP project. Would you like to be an assessor? And I immediately thought that this would be a really cool opportunity for me to actually go through and evaluate how well someone did at actually predicting phenotype from actual whole genome sequence. Again, they said that they put this on the website for fun, but if you think about it, this is really one of the grand challenges about what we're really trying to do in terms of genome interpretation. And I think my role here today is to talk a little bit about the submission. You'll hear actually from the submitters themselves. But I think my role here today is to be a bit of a cheerleader, to actually try to get you excited about trying to develop methods to actually predict phenotype. There's only 10 genomes that are available now, but we expect that there'll be more genomes available in the future and through the PGP for this competition, even perhaps before September. But long term, I would expect that we would have more and more genomes available to us. So today, as all of you know, there are companies and researchers that are out there that if you give them your genotype in some way, either the form of a biofluid or in the form of actually genetic data, they will come back and give you either risks to specific diseases or identify disease or phenotype-associated variants that are across your genome. And I think one of the goals of this competition, of this assessment, I did not say that, this assessment is specifically designed to evaluate what methods and what variants are good at prediction. Okay, so we'll start first with background on the assessment and challenge. We'll hear a video from Rachel Karchin's group, who was the lone submitter, about a little bit about how they did it. I don't know if they'll reveal everything since they are planning on submitting again this year, but we'll hear a little bit of those details. I will not reveal any of the answers here today, other than the assessment that we did, simply because this data is still available, and we hope that you will submit for the following year. All right, so who are the participants? The participants are 10 individuals whose genetic information is from the Personal Genome Project. The Personal Genome Project is, the mission of the Personal Genome Project is to encourage the development of personal genomics technology and practices that, and I'm quoting, are effective, informative, and responsible, yield identifiable and improvable benefits at manageable levels of risk, and are broadly available for the good of the general public. One of the hallmarks of the PGP is that they adopt an open consent model, and that participants agree to openly share their genomes and some of their phenotypes and traits, and this enables this project here. So what is the challenge? Well, if you go to the website, there are 10 PGP genomes that were data sets that were released. There is a number of different platforms that are available, Affymetrix, 500K, SNP chip results, exome sequencing results in a couple of different formats, and exome sequencing results in a couple of different formats, and you can go up and play with that data pretty easily. So some of the phenotype data, some of the phenotype data was available, and the goal was to use this data to predict the phenotypes not available on the public resources. Okay, so again, since this data set's being used again, I'm not revealing any of the actual traits, but we will evaluate the strengths, the weaknesses, and the challenges, and this will hopefully be useful for you, the submitters of this new round. All right, so what were the traits that were put on the website? Well, the first traits that they wanted to have predicted were binary, and they included a large number of things that you would expect to be highly genetic. Some were complex phenotypes, and some maybe are more Mendelian inherited phenotypes. And then we also had a couple of things that were highly environmental. For example, absolute pitch was one of the phenotypes that we evaluated. I don't know how many of you are aware of this. Absolute pitch is the ability of, the way it's technically defined, is the ability of someone to identify a note, basically what a note is played on a piano or off of a computer in about a second and a half, within a second and a half. If they can do that and identify what that note is, They have absolute pitch. They can identify this perfectly. There, in fact, are genetic studies that have been done to study absolute pitch. And as you would guess, you have to know what a note is. You have to know what music is to be able to tell you what that is back. So it's highly environmental as well. So we evaluated all these 32. There are also a number of numerical traits, some of which are very likely to have genetic components, and some which are completely environmental, including some that were maybe perhaps almost entirely environmental, including the astrological sign of the submitter, of the genome. So our first question when we sat and thought about this, at the time we were thinking about being competitors. We hadn't been asked to be an assessor yet. And we thought about some possible ways that people could go about doing and making predictions for this assessment. First, we thought, well, you could just go into the databases. We could go to OMIM, we could go to the HGMD, we could go into PharmGKB, we could go into the GWAS data in dbGaP, and we could identify all those variants, identify variants that were markers in the genomes, and then think about the rate that these phenotypes actually occur in the population, and then make predictions based on that. That was the first kind of approach that we thought people might be able to take. Another approach that people might take would be direct population-based approaches. Let's say you have a database of a large number of well-phenotyped individuals and a database with matched genetic information. We thought that, for example, companies like 23andMe or Navigenics have data sets like this. Large hospitals that are now collecting genetic data and connecting them with their EHR have data like this. And you could imagine developing methods that would look for essentially similar genotypes and make predictions based on what the phenotypes of those individuals were that had similar genotypes. I think this is a great idea. Again, we have absolutely no idea at this point, other than in literature, how well this works in practice. And then we could also imagine first principle-like approaches. We know that gene X may cause phenotype Y, and maybe gene X has variants that are likely to break gene X. And we see that those variants are present in both copies of the gene. We might say, well, then maybe that phenotype is present. We could find the data from other sources, or at the end, we could just simply guess. So I'm now going to show you a video that describes Rachel's group's submission. I think you'll find this very entertaining. And we applaud her considerably for, one, both doing an enormous amount of work, I think last summer, to make this method, to do really an impressive feat. And two, I hope to encourage you all to think about this problem in a real way. So here's Rachel's submission.
My name is Hannah Carter, and I'm a fifth-year graduate student in biomedical engineering with a background in electrical engineering.
Oh, hi, my name is Yunchin Chen. I'm a graduate student in biomedical engineering.
Hey, my name is Dewey. I'm a third-year biomedical engineering grad student.
My name is Jean, and I'm an undergraduate student at Johns Hopkins University.
Hi, I'm Shishelle Turner, and I'm a graduate student in the Human Genetics program.
Hi, I'm Rachel Karchin, and I'm the principal investigator of the Karchin lab. Hi, I'm Rachel Karchin from Johns Hopkins University, and thanks for inviting my group to present what we did last year in KG. I'm going to talk a bit about how we worked on the PGP challenge, and then I'm going to introduce Hannah Carter, who is one of the graduate students who played a really key role in this work, and she'll tell you about some of the details. This was our mission. We have 10 PGP individuals. We have their genotypes, at least we have partial information about their genotypes and their exomes. And our goal is to predict approximately 40 phenotypes. So once we worked out our rough ideas on the whiteboards, we started to really nail down some of the models. We decided to use a graphical model to do these predictions. Here's one of the early versions. We then simplified that first version, and then things got a bit more complicated with this version. Well, once we had these probabilistic models, we started writing down likelihood equations. Here's some of our mathematical thinking. Here's some more of it. Finally, here's the very simple model that we decided to work with. And I'm going to have Hannah step in here and talk to you about some of the details of how we did this work.
Since we were trying to predict the probability of each individual having these phenotypes, we needed to use a probabilistic model. For our model, we chose to use a BN2O network. Traditionally, these models have been used in medical diagnosis problems. It's a two-layer Bayesian network. The first layer, the blue squares, represent the causes, and the second layer, the yellow circles, represent the outcomes. An edge between a cause and an outcome represents a causal relationship between the two variables. In our model, the blue squares represent the genetic causes, and the yellow circles represent the 32 binary phenotypes that we're trying to predict. There were a number of genetic causes that we could have considered for our model, but due to time constraints in the data, we chose to focus on two types. First, we looked at significant variants. These are variants that were associated with the phenotypes we're trying to predict in a genome-wide association study. Second, we looked at affected genes. These are genes that harbor variation that is predicted to impair the activity of the gene's protein product. We treated these two types of variation different in our final model. First, we associated a prior probability with each of the phenotypes. In the first level of our model, we update our probabilities based on the significant variance from genome-wide association studies. In the second layer of the model, we update our probabilities based on the affected genes. We create one of these models for each of the 10 personal genomes in order to predict their individual phenotype probabilities. In order to estimate our phenotype prior probabilities, we looked at literature and tried to find population prevalences of each of the phenotypes. Where possible, we tried to control for age, gender, and population of the individual. Here we're taking a closer look at the first level of the model, the one dealing with the GWAS variation. In this level, for each individual, the blue squares are set to 1 if a specific GWAS variant is observed in that individual and left at 0 otherwise. The key component here is the lambda, the edge weight from the cause to the effect. The edge weight, lambda, represents the association, the strength of association between the variant and the phenotype. In this case, it's the penetrance, the probability of observing the phenotype given that the variant was observed in that individual. For the second layer of the model, we had to develop lists of genes associated with each of the phenotypes. We did this through extensive literature search, both manual and automated, and we also considered genes and regions of linkage with genome-wide association implicated variants. Finally, to get error estimates, we perturbed various inputs to the model to see how sensitive it was to those changes.
And now a word from our sponsor. While we have your attention, I'd like to tell you about a software package and database that is available from our group. The software is called CHASM. This is a machine learning method targeted at predicting significant missense mutations in cancer. But what may be of most interest to you all is SNVBox, which is a database where we've pre-computed approximately 90 predictive features at each codon position for every annotated transcript in the human exome. We hope that this will be of general use to people who are interested in developing machine learning methods and other kinds of predictors to predict the impact of missense variation. So thanks so much for inviting us, and we hope to see you next year at the KG meeting.
That was awesome. All right, so that was one submission. I hope you will take we'll take this to heart and think about submitting your own data set. Maybe you'll get a video of your own next ISMB. So how did we do the assessment? Well, we wanted, if those of you who were at the CAFA meeting yesterday, one of the things that we like to do when we do critical assessments like this is we like to have baselines. We like to think, well, what's the kind of we know what the prior probabilities are, but can we go a step farther and actually think, what is kind of, if this method that you've submitted didn't actually exist, how well could we solve that problem? And this was based on Stephen Brenner's suggestion. We used the Promethease tool from SNPpedia. Many of you may be familiar with SNPpedia. It's basically a Wikipedia-like project for SNPs, but they actually have an engine that for a little bit of money, they'll actually make annotations of SNPs that you submit. So a postdoc in my laboratory, Yanita Thursberg, actually paid the $20, downloaded the data, and actually ran that data on Promethease. And we compared some of those results to Rachel's submission. So first, we also took a look at the data. We looked at the binary trait. We had binary traits that we ranked by both a root mean squared deviation as well as an AUC from an ROC curve. We did these comparisons, and then we looked at individual traits. I'm not going to tell you which traits that they did well on, and the reason for that is because I believe their group is going to submit next year, and I would like to keep this as clean of a slate as possible so that you all can submit and not maybe perhaps have any idea how well you did. However, but I will say that for the binary phenotypes, they actually did somewhat well on some of them and made predictions. We didn't assess whether these were actually statistically significant or not, but they had relatively low RMSDs. Remember that some of the binary phenotypes are relatively rare and were unlikely to occur in that data set, so that might explain some of this. It was also some of the data was sparse. Only a few PG participants actually provided all of the data that they were looking for, and predictions, again, were largely the same across all the individuals. Interestingly, they got all of the astrological signs correct. So we also looked at precision recall and ROC curves. I think I'm just going to show the ROC curves now. They filtered only binary phenotypes. They filtered only, so we filtered the binary traits with only the predicted. The missed data points were excluded. We had 25 positives and 218 negatives when we took all this together. We combined all the predictions and plotted the ROC curves. This was done with the assistance from Predrag Radivojac at Indiana University. Thank you, Peja, if you're here. And we took a look at these, and we actually split out the overall ROC and the actual diseases that were in this list. And the ROC curve actually has some signal there, and we were happy to see this. The more rare diseases, the AUC was much lower. We looked at binary phenotypes predicted by SNPpedia and compared them, and SNPpedia basically only gives an X-fold increase or decrease of risk. We really don't have any idea how these, I don't personally have a good idea of how these methods work. Some of you may know. But we looked at them, and we actually did see some signal here. There was occasionally a case when Rachel's team and SNPpedia would agree on a specific individual in terms of risk. They're probably picking up on a SNP or a variant from a previous study. There are other cases when there was no correlation at all between the scores. This is rheumatoid arthritis. Sorry, the previous one was Crohn's disease. Type 2 diabetes, again, there were a couple of SNPs here that, in general, it wasn't a great correlation, but there were a couple of SNPs where it looked like both methods might, in fact, be picking up on something and suggesting that there is a marker in this individual for this risk. Again, another disease, coronary artery disease, not much signal at all. And not really a correlation. So finally, I guess I want to just sum up by asking, why is this so difficult? Well, obviously some of these attributes are highly dependent on environment. Some of these attributes are risk, and we don't really know whether the individuals will develop these traits in their lifetime. Doing the annotation from full sequence is also very, very difficult. and there's a lot of bioinformatics, there's a lot of basically heavy lifting that needs to go on in terms of the computation. And the coverage of traits by public tools like Promethease is very, very low. So I'm going to give some final thoughts. This part of the assessment is probably the dream, I think, of genetics and personalized medicine. Researchers are annotating genomes, companies are distributing risk variants, and the government, at least the U.S., is passing laws preventing use of genetic data to make it non-discriminatory, such as GINA. And even in this, we really have no idea how well we will do with this annotation. In this part of the KG conference, we will actually evaluate methods and our ability to identify phenotypes. And I think that's very, very exciting, and I hope this will be a very major growth area for this assessment. So I hope you submit next year, and I'd be happy to take questions if you've got time for them.